meeting? There we go. Good. So that's then the outline for the EEG course. Again, following roughly this idea of from first a general overview, which we'll also do for fMRI, and then following from the physics to the biology, and then discussing some fundamental things in terms of data acquisition and data analysis, again with the aim to expose you to all of this stuff, to familiarize you with the terms, and hopefully you learning some of the terms and definitions, not with the real my English. Always when I start again English, to start to teach English, I have to struggle a little bit with the words, with the real ambition to that you will learn how to do an EEG experiment or how to analyze EEG data after this course, more like you becoming familiar with terms. That's also actually another point on my teaching. So the way I see the first year is really for you, so I see it really as a theoretical year, because there will be you will maybe have some practical experience, maybe on your own or in Felix' class at the beginning of the second year and so on. But really it's about learning the theoretical background to all the practical things that you will then do afterwards for years to come. And that's also something how I think about university in general, because of course, outside of university and classes, there is no, I mean, you sometimes still talk a little bit about the theory, but in the end, all the world is doing applied stuff and practicing things. So this is why I think, personally, I think it's important to focus during the studies and in SCAN in the first year on the theory, because you will never have the time and the resources and so on to focus on the theory. So sometimes this leads, especially in statistical methods, that leads to the fact that people say, but can we also do it practically? And you will do it practically forever, but if you don't think about the theory and learn about the theory now, and I would just show you how to, for example, do some data analysis using some toolbox and so on, you will never learn the theory, right? Because later on you will basically do the practical stuff. Hence, this course also being on the theory side of things. And once you know the key terms and know theory, it's also much more sensible and fulfilling to do the practice stuff. So just be patient, and if you really can't wait to finally record your first participant or so on, just join a lab and they will happily let you record a lot of data. Definitely. Good, so that's the outline, and then let's start with the intro, where the idea is that we first go through EEG in a quick way, so that you basically know what we are talking about and what the content of the course is, and then we revisit everything and go more into details. So as I said, the course is based on this book by Stephen Luck on the event-related potential technique. It's a good book. It was kind of one of the first books that I found that is really written for kind of a cognitive neuroscience course. So there are, of course, many books on EEG, but some of them are really, so there's a Niedermeyer's EEG, for example, which is like, 1,000 pages and so on, and has many chapters, but is also very much on the clinical side. So that's mainly written by clinicians. And then there are older EEG introductory books which kind of don't deal with or don't think about EEG the way that we do these days, where we have computers and we have commercial EEG setups. So they are more concerned with essentially building your EEG system or something like that. So I think this Stephen Luck book is a good contemporary description of EEG. There is a new book, and I just made it smaller because hardly anything in the course will be based on this book. The nice thing about this MEG EEG primer is that, of course, it already in the title covers MEG as well. In the end, unfortunately, I didn't manage to read this book yet, but I skipped over the pages a couple of times. It looked, content-wise, very similar to what we have in the course and also to this book, but it definitely also discusses MEG a little bit more than we do. And the reason for that we don't go into that much detail on MEG is because next door we don't have the MEG machine, but we have the many EEG machines. And these books, you might encounter them. So you have these books, I assume. The other thing is that in this Google Drive folder, there's also a review by the authors whose name I now forgot, in Current Biology. It's called Primer, and this is a really nice article that summarizes, I think, in 4 or 5 pages the course. Of course, it's not going into much depth, but what I like about it is that it's from 2019. So it's a contemporary article that covers contemporary EEG in cognitive neuroscience, and it also demonstrates what I'm discussing here is not like how we did it in the 80s, but it's actually how things are happening as we speak. Yeah, so that's the literature. And the Luck book, I also have to say, and we will come back to that, it's nice. He has a specific viewpoint on certain things. For example, he has this whole somehow undefined concept of components in his head. And he and myself are not in line. So he talks a lot about components. What he really means are independent components in ICA, but that sometimes happens with these people that haven't done much of technical training. They then overshoot a little bit, and then everything is about components, because he's also of the kind of a group of researchers who use ICA a lot to analyze EEG data, and there are many ways to analyze EEG data, not ICA only, but because he's from that camp, there are certain parts in the book where he talks, for my taste, a little bit too much about components. So if you read the book and I'm telling you something, there sometimes might be certain contradictions, but if you detect them, just ask about it and we can discuss them more. And then it's due to the fact that, at least in this course, I don't really try to be from any camp, which is, I guess, not really possible. But and he's very strongly in the ICA camp. Yeah, good. So EEG. EEG is this thing where you put electrodes on participants' heads. This is kind of a current commercially available setup. Looks like this is actually also setups that we have here. So these little things are the electrodes. They have little lights in them which signal the impedance, something that we will discuss. And in general, you would put the EEG on participants by first positioning this white thing, this EEG cap, and then just plugging the electrodes in there. And after plugging them in there, putting in a little bit of a gel to have a good contact between the electrode head. So what you don't see here, we might see that later, is the little metal pin that is on top of this. That really then is the electrode and makes via this gel the contact to the scalp surface. And then if you show the electrical activity of these electrodes on a computer screen, this is then how things look like. So all these traces here are different electrodes. We will talk about the electrode naming and so on. One of these red lines here is 1 second. So you see that you have data for each of the electrodes within 1 second, and then we have many seconds here, and certain electrodes show certain stronger effects here. Not quite sure what this actually is. Could be something like ECG. But this is what I mean by EEG. So who has been a participant in this or recorded EEG themselves? Okay, and so the other people in psychology, so who has studied psychology but never encountered EEG? Okay. Yeah, so also can be the case. So that's EEG, how it looks like from the very surface. Talking about EEG and MEG, so just as a first impression, so the typical MEG setup used to, or I think it still is this, where you have kind of this magnetoencephalogram apparatus that you basically put your head in. You don't necessarily touch the surface, and within this thing here, so within this round thing, there are what are called squids, which are the detectors. So essentially you can also think there are like you have in the EEG, you have these electrodes that you put on. You there have magnetic field sensors that are in this machine. And it used to be the case that they really have to be cooled down close to absolute zero. And you have this big thing, and you have a lot of helium in there to cool things down. Now, quite novel, so this is from 2018, are MEG sensors which work at room temperature. They were actually not developed by these Boto people, but by some company, and they can be used at room temperature to record MEG. And in this paper, they discuss kind of a free moving MEG system. So here they have 4 of these sensors, which means that in terms of the traces that you get, which look very similar for MEG, but it's then the magnetic field, here's the potential. You have kind of 4 channels, and yeah, it's kind of something that they work on right now in certain labs. It's like always with a new technique, it's a little bit that people have high hopes that this now will finally solve all the remaining problems in cognitive neuroscience, like people thought when fMRI came around in the early 90s. Of course it will not, because in the end it's just again another kind of signal that you record, and you still need to make sense of the signal that you record. So the basic scientific questions that you will want to answer from these signals, like from these signals or from EEG signals, will remain the same. This is actually something that is my personal take. Of course, maybe we have at one point an MEG talk in the seminar series, then they might tell you a little bit more what the big difference between MEG and EEG is. This shows, that's actually a supplementary figure from this Nature paper, and they recorded both these OPM MEG, which is this MEG in kind of at room temperature, and they recorded EEG, and then they did an experiment where people moved their fingers and then looked at what is known as a time frequency analysis. We will actually talk about what this is at the end of this introduction. For if you don't know what that is, doesn't really matter right now, but what comes out of these analysis are these kind of plots where you have 0 here and then it's over time, and you see some frequency changes over time. And this is the result from EEG electrodes, roughly where also these MEG electrodes are for the EEG here and for the MEG here. And so you can compare essentially this and this, and this and this and this and this and this and this. Looks a little bit different, but also looks quite similar, I would say. So it's blue here, and then it gets red, and it's blue here, and then it gets red. And here it's blue here, and it's blue here, and then it gets red, and then it gets red. So in terms of the signal and the end and the result of your data processing, that's how it looks. Of course, one can, so people also combine MEG and EEG recordings, and then if you do that based on some biophysical model that, for example, says, okay, what you have in the signal is more on the deep source, is more from the deep sources, and what you have in the MEG is mainly from tangential sources, so being tangential to the cortex, then you can make more of that. And also, can do interesting kind of signal integrations. Right now, my personal take is if you do basic analysis of MEG or EEG data, yes, they are different physical units. Yes, they reflect different things. But of course, I mean, this is always implied. Electrical activity is always accompanied by magnetic activity and the other way around. So if something electrically is happening, something magnetic is happening, as the physicists in the room will happily tell you. And if not, just intervene. So it's similar, and as these things always happen together. So this is a little bit of the kind of very brief justification that we will focus on EEG in the following. And not do much with MEG, because in the end it just detects the other side of the coin, if you want. If you see one side is the electrical activity, and then you have the magnetic activity, and if you record this or the electrical activity, it's roughly showing you the same thing. Of course, again, if somebody's really doing MEG, they will have more arguments, and hopefully more in-depth arguments for using MEG. But not necessarily. So you can also have the case that a cognitive neuroscientist walks in the room and shows you their new MEG study and how things are different between being conscious of something and not being conscious of something in MEG. And then if you ask them, so you used MEG, so why did you use MEG and not EEG? They would say, well, we have this MEG machine there. And then if you ask, so do you think something would be different in EEG? They will say, don't know, or maybe not. So depends always. So it doesn't really mean that if people use a certain technique that they use this for a very good reason. Maybe it's just because the lab uses this technique and they use this. Anyway, there's nothing wrong about MEG, but it's also not that different from EEG.
As far as I know, MEG is quite expensive compared to EEG, and they just show similar activity.
Exactly. Yeah.
Why we should use it? Why they use it?
Yeah, and this is then where the science starts. So yes, I agree. So the definitely this kind of MEG is quite expensive, and this is why a lot of universities also don't have these MEG machines. This is also why there's some interest in this. This is cheaper. And then, as I said, there is some hope that because it's not by means of volume conduction, but because it's directly there, the magnetic field, that it's a little bit better in terms of spatial resolution. But then again, being only sensitive to certain aspects of neural activity as opposed to EEG. So, and I agree in terms of the costs. EEG is these commercial systems. They are not cheap, but they are definitely cheaper than this, and this is obviously more experimental. And EEG is definitely a lot cheaper also than fMRI. Hence, I am mostly interested in EEG actually, because it's cleaner, it's less harmful if compared to MRI, and it's I mean, we will talk more about the fMRI versus EEG, but it's it's not harmful, or there are no you don't need special cautions, and mainly it's cheaper, and it does all the same things. So from my perspective, we could just go away with EEG unless we have very good reasons to do MEG, and then we have to go much more into details than doing a time frequency analysis already. I mean, we have to do more than this, which is a kind of a standard analysis in cognitive neuroscience. Yeah. Good. So that was contemporary MEG, EEG. EEG has been around for 100 years almost. As we will see at some later point, neurophysiology, so recording electrical activity in animal neural systems, had been around also at in the late 1900s. But EEG and the possibility actually to record from the scalp activity that relates to the brain has been only reported first by Hans Berger in 1929, I think in Jena, but was not really accepted by the scientific community of his age, because apparently he was also interested in parapsychology, so he had certain interests that didn't resonate too well with the scientific community, so they didn't believe that when he put the EEG on human scalps that anything of course people saw that there's some electrical fluctuations, but the thing was that they didn't believe that it had to do anything with the brain. Right, so it was not controversial whether there's something happening in the electrode, but whether this relates to the brain. And the initial discovery was the alpha rhythm. So does anyone know about alpha? What alpha is?
That's the usual range for the resting state.
Yeah, and do you know any about the frequency range of alpha? Yes. Yeah, exactly. So 10 Hertz is the typical alpha, and alpha usually shows up when people close their eyes and start being a little bit sleepy. I wonder whether there's some alpha in here. So if you see this. This where you see a little bit of a pattern in the traces. This is alpha. We will see more alpha later, but this is what Hans Berger first reported, and it was also what was weird about it was, of course, that you saw more activity when people actually closed their eyes. So that was also very counterintuitive that when the brain stops processing stuff, you suddenly should see activity. But then this was found again, and then people started to believe that more in the 30s, and then already in the late 30s there were reports of sensory event-related potentials. I assume this must be very strong, or I'm not quite sure, actually I don't know the paper behind this, whether this was already based on averages or whether this was, I guess it must have been on averages, because people are still fighting for single trial event-related potentials these days, as some people might remember. No, you were not there. Were you there when Gabriel Curio gave a talk? No. With his single trial event-related potential. But anyway, so event-related potentials, which we will talk about at length, also have been around since the 40s or 30s. but mainly with relation to sensation. And then in the 60s, the cognitive event-related potentials and took off in psychology, including the P300. Which we will also discuss at length. And this basically, so these works in the 60s that related EEG activity to cognitive phenomena. This then gave rise to EEG being used in cognitive neuroscience, increasingly, and also in psychology departments, essentially. And of course, this whole EEG endeavor, like all kind of data analysis endeavors, benefited a lot from the digital revolution, so that there were computers more readily available. And then since 1990, it's actually one of the tools in cognitive neuroscience, I think, or it feels a little bit like, of course, there was much hype around functional magnetic resonance imaging in the mid-90s to maybe the mid-2000s. So after then everybody had done their first fMRI study and they figured that they still don't know how the brain works, and that also fMRI has its weak points. EEG became kind of had a little bit of a comeback, if you want, and now is kind of on par, I would say, in popularity with fMRI and MEG. And I think that's a little bit of the history. So I think this is actually one of the few slides that are from the MEG-EEG book, because they such a slide doesn't exist in the Luck book. So this talks about the frequency bands. So and also makes the point that kind of looking at frequency power had been around, or has been around in EEG and MEG forever. So it basically, sometimes you think that doing time frequency analysis or doing Fourier analysis of EEG data is very recent, but this is also actually how the whole thing started. Like I said, alpha was already described by Hans Berger in 1929. So that's 10 Hertz activity. Lower fluctuations, which are referred to as theta or delta, have been reported later, of course, with respect to sleep. So delta activity. Beta has already also been described by Hans Berger, and beta is currently also of interest to many cognitive neuroscience people. And gamma has also already been described in the 40s, and was at one point around briefly before I entered, I think, cognitive neuroscience. Gamma was consciousness. I hadn't heard much about gamma and consciousness recently, but these are these frequency ranges, so oscillations at a specific frequency and timing of oscillations that just come with EEG and MEG, and it's good to know their rough frequency bands, because people. So what cognitive neuroscientists often do is that they will say, working memory correlates with beta activity. That's a typical cognitive neuroscience claim, so it's good to know roughly what people mean by that. If you really want to know what they mean by, if they say working memory correlates with beta activity, you have to look into their papers, how they do their analysis, which frequency range they really mean. Do they really mean everything between 40 and 30, or did they focus on 20 because for 20 it actually correlated and for 21 it didn't correlate? Something like that. It's unlikely because there's some smoothing over our frequency space, but so it's good to know these things. They've been around forever, and we will talk a little bit more about how you actually get to these frequencies and what it means when we get to a little bit of Fourier analysis in this course. Questions about this delta, theta, alpha, beta, gamma? Delta? Alpha? No. Good. So then to get started a little bit on EEG terminology, because after all, also one main thing in the whole SCAN course is that you learn a new language, meaning that you learn new vocabulary, words that have a meaning to these kind of people that deal with cognitive neuroscience, and you also learn how to so you learn some vocabulary and semantics of these things, and you also learn some grammar, so that, for example, evoked potential doesn't go well together with fMRI. So if you go somewhere and say, I recall an evoked potential with my fMRI, then people will be like, N400, that doesn't work. In cognitive neuroscience. Outside of cognitive neuroscience, people will be, you must be pretty smart. And sometimes in cognitive neuroscience as well. So, new terms. Evoked potential. What evoked potential means that these are electrical potentials which are evoked by sensory stimuli. So they come in different flavors together with the sensory modalities that mammals have. For example, there are things called visually evoked potentials, which are potentials that are evoked by visual activity. They are somatosensory evoked potentials, which are potentials evoked by the sense of touch, so somatosensation. There are auditory evoked potentials, which are of course then evoked by audition. There are also gustatory evoked potentials, but of course visually and somatosensory evoked potentials are used primarily in experimental science, because of course it's much easier to show to a participant a visual stimulus or apply some brief touch stimulation, which is already harder than doing visually evoked potentials or visually visual stimuli, than to do something like where people taste something. Then, because how to administer the stimulus and how to time it and so on is a little more complicated. So this is what evoked potentials are. Evoked potentials, however, are also interchangeably actually referred to as event-related potential. And this is a little bit about the terminology here. So this is from the Luck book. Since cerebral processes may be related to voluntary movement and to relatively stimulus-independent psychological processes, the term evoked potential is no longer sufficiently general to apply to all EEG phenomena related to sensory motor processes. And some of them may serve as time. And on the other hand, you also not necessarily always average data based on a stimulus, but also on some other kind of event. Hence, the term event-related potential was proposed already in the late 60s to designate the general class of potentials that display stable time relationships to a definable reference event. And this is why this whole book is called Event-Related Potential Technique. And also we these days talk actually more about event-related potentials than evoked potentials. However, if you're dealing only with a visual stimulus, then you might still call this the visually evoked potential, same for the SEAP. You can also call that a visual event-related potential. But so this name event-related potential is there for a reason. It's somewhat synonymous with the term evoked potential, but evoked potential being a little bit outdated. That's what I said. Good. So how do these things come about? So this is a typical EEG design. So the typical setup works that there's a participant. Here you see the head. They watch a computer screen, usually if you have visually evoked potentials. And on this computer screen, certain stimuli are applied. And in this case, because it's an so-called oddball design, the only thing that is displayed are X's and O's or circles. So you have these X, and then you have two O's being presented, and they are presented every 1.5 seconds. Yeah? So this X shows up, stays maybe on, maybe until here, or maybe a little bit longer for 1 second or so, and then after a short break, a new X comes on, or an O comes on, and then again an O comes on, and X comes on. Whenever this comes on, there is actually a signal sent from the computer that drives this stimulation to the computer that records the EEG. So the person sits in front of the screen, has the EEG on. The EEG, the electrodes are connected to a computer so that the electrode activity is recorded, and the computer that runs the stimulation also is connected to the recording computer, so that whenever something happens on the screen, this is also recorded together with what is shown here, namely the potential curve of one electrode. And here you see that EEG activity is recorded basically just continuously. So you just, as we saw earlier, you just have these traces that in each, usually, so it depends on the sampling rate, but usually every 2 milliseconds there's a potential measurement made, and then all these measurements are just put next to each other in time, and you get this trace. And yeah, this just shows some electrodes. It's not really important right now. The important thing is that this kind of raw data recording is not what you then look at and what you call an event-related potential, because what the event-related potential is basically the time course of the potential after averaging. Yeah. So the important thing is that if you look at event-related potentials, that you cut out a given time segment after a stimulus onset, usually or often an external stimulus, sometimes an internal stimulus, whatever that may be. And then you take these recordings for each of the single trials and you average them together. And this what you get out of the averaging, this would then be called an event-related potential. So you average over multiple trials, usually. So this is what is shown here. So now think that you cut out this, where this block is, and the different blocks. One thing that is shown here. So it looks like a continuous thing, this thing, so the trace. But if you really look closely, of course, it's discrete because it acquires one measurement every 2 milliseconds. So this is what this is supposed to show. That, of course, the signals that we record are always discrete. So whenever you deal with continuous functions in math, they are just approximations of discrete things, if you want. Now, like I said, you cut out from one electrode what happens after onset, and we had 2 X's first, and then 2 O's, and then 2 X's. And now if you average together everything from the X's and everything from the O's, this is what you get. And this is what is then usually referred to as an event-related potential, of course, at a given electrode site. So this would be, in this case, this electrode here, which could be something like FZ, for a given electrode, and then also for a given stimulus. So this would be the average of all the X stimuli at electrode FZ, and of course for a given time window after stimulus onset. So the stimulus onset would be here, and then this would go until, I think here, maybe 800 seconds after, 800 milliseconds after stimulus onset. So that this whole thing here then extends, there we have it, it extends 800 milliseconds. Yeah? Meaning here there always was a stimulus, and this is the average response for this electrode for this condition. And this is then what is referred to as an event-related potential. And then these event-related potentials have different peaks and valleys. And whether they are peaks and valleys, that depends a lot on which electrode you're looking at. This is also, I think, nicely discussed in this current biology review, because classically, they didn't used to be 64 electrodes or 128 electrodes like we have now, but they used to be 3 or 4, well, I guess 4, and that were at certain places. And at these places, then some neural activity would always show up as a positivity. So this introduced into the EEG literature the notion of positive amplitude fluctuations, which are abbreviated with P, and then the first positive amplitude fluctuation would be P1, the second one would be P2, and the third P3. And same for the negative deflections, that would be N1, N2. And this is somewhat confusing if you ever really look at real EEG data, because, for example, the that's actually not the best example, but let's say the P100, which is a typical amplitude fluctuation in response to a visual stimulus, this will be positive in certain electrodes, but at the same time you have a negativity in other electrodes. Because, as we will see later, this is due to some dipolar activity that at one part of the scalp makes everything negative, and on the other side makes everything positive. So there's this nomenclature of, for example, saying P300, but the P300 has never been a positive amplitude deflection in a frontal electrode. It's only positive in a occipital, so an electrode at the back of the skull. And this is due to the fact that when people first described these amplitude fluctuations, they only had like an electrode there where they were interested in, and there it was positive, hence they call it a positive thing. But that's something to keep in mind.
What is negative potential concretely in neural terms?
Oh, we will talk about this. So we will talk first about potentials from the electricity perspective, and then we will talk about the biological thing. For now, it's just being above or below zero in these curves, and then that's where the nomenclature comes from. Another thing, actually, from my perspective on this whole thing about naming these peaks and valleys, is that there is a whole literature on the N2, or the P1, or there's a whole literature on the P300. Or the P3. Actually, that's another thing about this nomenclature. Sometimes they're referred to which peak they are. The other one is the other nomenclature is at what latency they are. The problem with the latency is that there is some variation in the latency. So the P3, sometimes for certain stimuli in certain regimes happens at 250 milliseconds. So is it then the P300? No one knows. But it's the third positive peak. The other thing is that what has happened is that some people really focus on like the N2. And if you ever do a real an EEG experiment yourself and you compare two conditions, you will see, as you also see here already, that these ERPs at a given electrode site, let's say, they don't, for different conditions, they don't differ only in one of these. Usually they differ in at least, so if you have condition effects, specifically cognitive condition effects, they will affect a lot of things after, let's say, 100 milliseconds. maybe over conditions, the early are very similar, but then they start to diverge. And if you now have people applying classifiers to EEG data, they will sometimes then see deviations fairly early on, and this will be kind of the novel result. But it's not. I mean, if you look at if you just look at the potentials in electrode space, you will see that it's never the case that over different conditions everything is the same, but one of these peaks is different. It doesn't happen. So this is why I'm also, so kind of models that we develop, they are interested in modeling the whole event-related potential time course. So basically have neural systems that create this entire thing and not cutting out stuff and focusing on one of these amplitudes, because it's really hard to pinpoint, because it's of course many neurons, many cortical areas that contribute to this. Good. So this is a little bit just as a warning for in terms of these labels P1, N1, and so on. Good. And let's see what the next slide is. Maybe we do that until Miriam comes, because this is just also one thing that in EEG people used to plot negativity upwards, so that on these kind of graphs where since school you have learned that what's going upwards is positive and what's going downward is negative. In EEG, they used to do that differently, so that negativity is upwards. And it makes no sense whatsoever. And Luck and I agree on this. And I don't do it, he doesn't do it, and we shouldn't do it because it's just completely weird. And you still see it sometimes that people come to a CCNB seminar and show EEG activity. Of course, negativity is plotted upwards here. Why? Are you outside of the normal world? What is going on? This we discussed, and this we also discussed. It just says about the P's and the N's and the naming conventions. But please plot your EEG activity with positivity upwards like the rest of the world. Good. I think I want to stop here, because we are already at 45 minutes and Miriam should come in any time. And then we will start again tomorrow at 9 o'clock with continuing this introduction to EEG and then starting to go into the details of the electrical foundations of things. Maybe one last thing, some of these slides have pictures on them. And some of the slides have text on them. The slides with the text on them, this is always good to memorize. So this is more not me not knowing that I shouldn't show you pictures so that you are happy. This is more that I condense what you should memorize. The stuff with the pictures, it's hard to memorize, but it's nice, sometimes nice to look at. Good. Any questions? Anything for today? No? Good. Then I see you again tomorrow at 9, unless you want to do something else.